This lecture investigates the second type of cell division, meiosis. If I asked you where babies come from, you might not think it has anything to do with meiosis. However, meiosis is necessary for sexual reproduction. Some organisms can reproduce by asexual methods, such as this hydra. It can reproduce an offspring by budding, in which a genetically identical individual grows on the parent. These trees can also reproduce asexually. All these trees in this stand are clones, as they all sprouted from the same roots of a single individual. But few organisms reproduce solely by asexual means. Almost all life has the ability to reproduce sexually. In sexual reproduction, gametes such as eggs and sperm are formed during meiosis. When the gametes fuse, a fertilized egg, or a zygote, is produced. Meiosis produces haploid cells from diploid cells. Diploid cells contain homologous chromosomes. These chromosomes occur in pairs which come from the parents. For example, one chromosome from each pair comes from mom, while the other comes from dad. A pair of homologous chromosomes are similar in length, shape, and centromere location. They also have the same gene sequence. Somatic cells are diploid. Somatic cells are all the cells except the gametes. This diploid cell has 8 chromosomes, or 4 pairs of homologous chromosomes. Haploid cells contain only one member of each homologous pair. Gametes are haploid and are produced by diploid gametocytes through meiosis. So this egg and sperm would only have half the number of chromosomes as a diploid cell. If human somatic cells have 46 chromosomes, how many chromosomes would we find in the gametes, the egg and sperm? Right, 23. And when a human sperm fertilizes a human egg, a diploid cell is produced. Sexual reproduction involves the production of gametes. When these gametes fuse, a new individual is produced. Half the genetic information comes from the father, while the other half comes from the mother. So if we looked at one of your somatic cells, we would find 46 chromosomes in the nucleus. Of these 46, 23 came from your mother and are considered maternal chromosomes, while the other 23 came from your father and are considered paternal chromosomes. But these chromosomes are really homologous pairs. Each maternal chromosome has a partner paternal chromosome, which is the same shape, size, and contains the same sequence of genes. For example, if we looked at a certain loci, or location, on this maternal chromosome, we would find the same gene on the paternal chromosome in the same loci. Meiosis involves a diploid cell going through 2 stages of cell division to yield 4 haploid cells. Let's look at the life cycle of this dog. The dog, like many other organisms, is diploid. Its somatic cells contain homologous pairs. Through meiotic cell division, the dog produces gametes, sperm in males and eggs in females, that fuse to form a diploid zygote. Mitotic cell division and differentiation of the daughter cells produce an embryo and, ultimately, a sexually mature dog. Let's look at an overview of meiosis. This simplified cell has a pair of homologous chromosomes at the G1 phase of interphase. In a human somatic cell, we would see 23 pairs of homologous chromosomes. When the chromosomes replicate in the S phase, we get a homologous pair of replicated chromosomes. The first cell division, meiosis I, separates the homologous chromosomes. And haploid cells are created at this stage because each cell contains only one set of the homologous pair. The second cell division, meiosis II, separates the sister chromatids. At the end of meiosis, we have 4 haploid cells. The events that occur in meiosis are similar to mitosis, so let's look at the differences in detail. During prophase I of meiosis I, the homologous chromosomes pair up or synapse. While they synapse, an event called crossing over occurs, in which non-sister chromatids exchange genetic material. This results in genetic recombination, as the chromatids now have a new combination of maternal and paternal DNA. Here's our homologous pair of replicated chromosomes. Let's say the maternal is red and the paternal is blue. Each duplicated homologous chromosome is composed of 2 sister chromatids, which are identical to each other. But a maternal chromatid and paternal chromatid are not completely identical. While they have the same gene sequence, there might be different forms of those genes. So these are called non-sister chromatids. During prophase 1, the maternal and paternal homologous chromosomes pair up to form a tetrad and are held together by the synaptonemal complex. When this occurs, we say the chromosomes undergo synapsis. Here is a diagram of a pair in synapsing homologous chromosomes and prophase 1. The synaptonemal complex is composed mainly of protein, and while the homologous pair synapse, another event can occur. Crossing over. This is when genetic material is exchanged between non-sister chromatids. Crossing over can occur in multiple sections of the chromatids. The point where crossing over occurs is called the chiasma, or chiasmata if there are many regions of crossing over. In metaphase 1, the tetrads line up in the midplane of the cell. In anaphase 1, the synaptonemal complex breaks down and the homologous chromosomes separate. In telophase 1, the new nuclei created are haploid, as they now contain only one set of homologous chromosome. But each chromosome is still composed of chromatids. The second round of cell division is very similar to mitosis. In metaphase 2, the chromatids separate to form 4 haploid cells. Let's look at the whole process using a gametocyte from the fruit fly Drosophila melanogaster. Here's the diploid cell of a male fruit fly. How many chromosomes does it have? Yes, 8. The cell grows during G1. Chromosomes replicate during the S phase, and the cell prepares to divide in G2. Remember, in prophase 1 the spindle begins to form. Homologous chromosomes synapse and form tetrads. This is also when crossing over can occur. Crossing over does not take place in male fruit flies, but it does in most organisms. In metaphase 1, the tetrads line up at the midplane, and the homologous chromosomes separate in anaphase 1. In telophase 1, the haploid nuclei form, and cytokinesis creates 2 daughter cells. In prophase 1, the spindle forms and attaches to the chromatids. In metaphase 2, the chromatids line up at the midplane. In anaphase 2, the chromatids separate. In telophase 2, the nuclei reform. And cytokinesis creates 4 daughter cells. Remember, 4 haploid cells are created by meiosis. Now you might wonder why you don't look like your siblings, even if you have the same mother and father. This can be explained by 2 events during meiosis. The first is crossing over. When those recombined chromosomes are created, genetic variability increases. This is because we will have 4 different types of this chromosome. 1 that just has the maternal DNA, 2 that have a combination of maternal and paternal, and 1 that just has paternal. The other event is homolog shuffling. This is the random orientation of the tetrads during metaphase 1. Let's take this cell. If the homologous chromosomes are arranged this way, we will have this combination of maternal and paternal chromosomes in metaphase 2. At the end of meiosis, we end up with 2 cells that have 1 maternal and 3 paternal chromosomes, while the other 2 have 3 maternal and 1 paternal. But let's change the orientation of this second set of homologous chromosomes. There. Now the maternal chromosome is facing the pole to the left and the paternal is facing the pole to the right. Let's find out what the resulting haploid cells will look like. So with that one change, we have 4 haploid cells with 2 maternal and 2 paternal chromosomes. In addition to the events in meiosis, it is random concerning which 2 gametes will fuse to produce the diploid zygote. So you are not like your siblings, and no one but an identical twin has your DNA sequence. Homologous shuffling, crossing over, and the fusion of random gametes results in there being over 24 trillion different combinations for humans. And that's what sexual reproduction does. It introduces genetic variability for each new generation. So make sure you know the differences between mitosis and meiosis for your exam. This diagram starts with the parent cell with a diploid number of 6. That's what the 2n equals 6 means. Mitosis goes through one cell division and produces identical diploid cells. while meiosis goes through 2 cell divisions and produces 4 haploid cells.